Men's bodies, part 2. In this lecture, we're going to be covering males' bodies, but we're going to be focusing on male reproductive physiology. We're going to discuss some of the organs that we had talked about in our previous lecture in more detail in our lecture today. So let's start first. Now that we've gone through all of the various anatomical parts associated with the male body, let's talk about how an erection occurs. Well, ultimately, an erection is influenced by the autonomic nervous system. And the autonomic nervous system is nervous tissue of the peripheral nervous system. Now, in this diagram, we have the central nervous system. That includes the brain and the spinal cord. However, it's important to note that the central nervous system, the brain, and the spinal cord is not the only type of nervous tissue in the body. The spinal cord has nerves that are going to branch out and innervate various areas of the body. That's important because that helps control our physiological responses. It is the autonomic nervous system that is part of the peripheral nervous system that branches out from the spinal cord, and this ultimately innervates the genitalia as well. So that is important for stimulating an erection. Here we have areas of the central nervous system where these nerves branch out. And when they branch out, they're going to target the penis, either to become flaccid or to stimulate it in order to be active and erect. So let's elaborate on this some. In order for an erection to occur, a stimulus or a sensation results in an electrical impulse being sent to the erectile tissue, and that erectile tissue is innervated with nerves. Remember this cluster of nerves that are really in high concentration in the glands of the penis. They're called genital end bulbs. Well, upon stimulation of the penis, some kind of stimulus, what's going to occur is an impulse is going to go from the genitals, which are especially sensitive due to the high concentration of genital end bulbs, and that impulse will go from this peripheral nervous tissue to the central nervous system, to the spinal cord. And then this is going to evoke a response right back. Now this response that goes right back, notice, this is going to muscles of the pelvic floor. This is also going to the genital area. So this impulse is going to stimulate the release of a neurotransmitter called nitric oxide. Nitric oxide is a neurotransmitter that's going to cause arteries to dilate. Now when arteries dilate, that's going to enable blood to pump into the penis. And during this process, veins will also constrict. So blood goes into the penis, but not much blood goes out. That's going to result in tumescence or an erection. The spongy tissue within the corpora cavernosa will engorge because of the high concentrations of sinusoids. Those sinusoids are the collapsible spaces within the corpora cavernosa. So as blood continues to fill into the penis is going to increase in length, increase in girth, and because not much blood is escaping, this will continue to occur until a male reaches a full erection. This is important because this helps penetrate the female reproductive tract. Now, upon continuous stimulation, males often experience an orgasm. And an orgasm is accompanied by 2 phases. These phases are emission and expulsion. During the process of emission, the prostate, the seminal vesicles, the vas deferens undergo contractions. And when this occurs, the contractions are going to force semen into the prostatic urethra. So this prostatic urethra we're going to talk about in a little bit more detail because this is important during emission and expulsion. The prostatic urethra is essentially a part of the urethra that goes or passes through the prostate gland. Now, urethral sphincters are going to close, and that's going to trap semen in the urethral bulb. The urethral bulb is part of the urethra that passes through the prostate gland. However, I've made a distinction here that the urethral bulb, this is the area of the urethra in the prostate that expands during emission. So during this process, it's important because this urethral bulb has semen there, has fluid there that accumulates, It's trapped and it's under pressure. This is also going to prevent any leakage of urine from entering into semen, which could contaminate or even kill some of the sperm within the semen. So now that we've talked about emission, let's see exactly where this occurs. Now, here with our diagram, we see that, well, here we have the scrotum. During an orgasm, the scrotum is going to tense. The testes will be a little bit closer to the body during an orgasm. Here we have the testes. The testes have important cells, interstitial cells. We're also going to talk about some Sertoli cells that are inside the testes today. Here we have the epididymis, where when sperm is produced in the testes, the sperm is stored in the epididymis. Here we have the vas deferens. And then the vas deferens is going to merge with seminal vesicles into the ejaculatory duct. And then here, right here, we have the prostate gland. And then this is the urethra. The urethra merges with the urinary bladder, and ultimately it's going to merge from the ejaculatory duct. So this is the urethra that is eventually going to make an opening to outside of the penis. So during emission, the prostatic urethra is going to expand. So remember, the prostatic urethra is this part of the urethra inside the prostate gland. And when this expands, that's going to accommodate more fluid. Now, to ensure that that fluid does not go into the bladder or seep out prior to ejaculation, it's trapped by muscles. There's a muscle called the internal urethral sphincter that's going to prevent any urine from leaking into the semen. And here is the external urethral sphincter that is going to be temporarily contracted. All of this fluid is subjected to pressure. And during emission, males can feel when this occurs. Emission is the point where a male experiences he's going to ejaculate. It is inevitable. That is usually when fluid begins to go from the seminal vesicles, sperm from the vas deferens, fluid from the seminal vesicles, and fluid from the prostate all pull, resulting in pressure that can be felt. After emission. Emission lasts for just a couple of seconds, maybe anywhere from 2 to 3 seconds. Then there's expulsion. Expulsion, sometimes referred to as ejaculation. They mean the same thing. During expulsion or ejaculation, this occurs after emission, where pelvic floor muscles that surround the urethral bulb and root of the penis are going to ultimately contract. The initial contractions are the strongest, and that's important. Sequential contractions are weaker, and the duration between these contractions is about 0.8 seconds. This explains why when males ejaculate, semen is expelled in spurts. And when it's expelled, the vast majority and force of semen is released during the initial contraction. It's released, and then the next contraction, well, there's not as much fluid. And then the contraction after that, there's even less fluid with less force. That's because muscles are contracting, and that is why semen is expelled in that fashion. The first contraction has the majority of the sperm within the semen, the majority of the volume of the semen, and it is the strongest contraction where the vast majority of the content from the semen is going to be expelled out of the urethra. Here we have a diagram demonstrating expulsion. So in this diagram, I've added this extension of the urethral bulb indicating that this is expanded and it's under pressure. Notice that currently at this point, expulsion is about to begin, but there are these muscles here. These are flanking the urethral bulb, ensuring, again, that semen is under pressure. So external urethral sphincter is going to relax. And when that happens, when this relaxes, there's going to be contractions. And contractions are going to occur at the urethral bulb, at the pelvic floor muscles, and at the root of the penis. And this is going to enable the semen that was once trapped inside the urethral bulb to now escape. And it only goes in one direction. So it's pushed out forcibly. And it's expelled in spurts. The first one, second one, the third one, and then maybe just drops after that. So there's anywhere from 3 to 15 rhythmic contractions, and during the process of ejaculation or expulsion, this takes approximately 15 seconds. So what exactly is released when males ejaculate? What's in semen? Well, of course sperm is, but there's a whole lot more ingredients than just sperm. Semen is fluid containing alkaline secretions from the prostate. As we had discussed earlier, these alkaline secretions are basic secretions. They're going to help neutralize the acidity of the vagina. So alkaline secretions from the prostate, seminal vesicles, and small amounts from the bulbal urethral bulb are going to help protect the sperm from the acidity inside of the vagina. This also contains fructose. Fructose is a sugar. This sugar is important because sperm require this sugar in order to propel themselves to find an egg. There's salt. That's important, so sperm are at an optimal ion concentration. That's favorable for cells. Buffers, that's going to minimize any drastic changes in the environments in which they live. Any drastic changes like changes in acidity, for instance. Antioxidants, this is going to help protect sperm against air. And this will also help protect sperm against oxidative free radicals they can produce in their voyage on the way to fertilizing an egg. Also enzymes. Interestingly enough, when semen is ejected, semen can be chunky. It's because there's some clotting factors there that are going to help help sperm remain together initially. However, if semen is released and it's kind of chunky and viscous, well, over time, there are other enzymes that are going to break up some of those clotting factors. That's important, so sperm can stay together when it enters into the woman's reproductive tract, but enzymes can then break those clotting factors so sperm can be released and they could swim on their heroic voyage to fertilize an egg. Well, males produce about 100 to 400 million sperm per day. Every day. We learned previously that women have a set number of follicles, and every month one follicle grows and emerges and is released. Females release one egg a month. Much of their reproductive cycle is dedicated to developing that one egg so it can be released a month. Males produce anywhere from 100 to 400 million sperm per day. Sperm are stored for weeks, anywhere from about 10 to 60 days. Interestingly enough, males release anywhere from 100 to 700 million sperm in a single ejaculate. That is profound. Because in males, their strategy is producing a high number of cells, and very little is devoted to each one of these sperm cells. Likely the most competitive or fastest sperm cell may get to the egg first. In females, the strategy is different. It's about the quality of one egg as opposed to producing many of them. So there's anywhere from 100 to 700 million sperm released during a single ejaculate. On average, it's about 250 to 500 million sperm, one ejaculation. Now, the sperm within an ejaculation can vary. It could vary depending on one's age. The quality of sperm begins to decline. And sometimes males produce less sperm as they're older. Certainly things like chemotherapy or drug use, poor health, those things could also affect sperm production. Duration of arousal, this could affect the amount of fluid that's within the semen. after multiple ejaculations, the volume can diminish. So if a male ejaculates, and sometime later, let's say 30 minutes to an hour later, he ejaculates again, that male has depleted much of the fluid that was used to protect the sperm. So although there may be many millions of sperm there, that fluid may decline. And only 1% of volume in semen is sperm. The vast majority of the fluid released during an ejaculation is fluid from seminal vesicles and the prostate. Very little of that fluid contains actual sperm cells relative to the total volume. And that's staggering, being that there's 250 to 500 million sperm per ejaculate. That should give you an idea of how small these sperm cells are. So now that we've talked about semen, we've gone through the ingredients in semen. We've talked about the process of males releasing sperm in semen through emission and expulsion or ejaculation. Now let's talk about the process of sperm production. And let's see how hormones coordinate this process. The process of sperm production is called spermatogenesis. This is the process of sperm production via meiosis. Now this occurs inside seminiferous tubules of males. And within these seminiferous tubules are very important cells called Sertoli cells. These Sertoli cells are very important cells inside the seminiferous tubules, and they're going to aid and stimulate spermatogenesis. It takes about 64 to 72 days for division and maturation of sperm cells. When spermatogenesis occurs, spermatogenesis begins at the outer walls of the seminiferous tubules. When this process is complete, this results in 4 viable sperm cells. And the testes constantly produce sperm. They don't go through cycles like a female does when she's producing an egg. Males have checkpoints that could stimulate the release of hormones, but they don't have cycles like women do. They don't have a time of the month where concentration is high and then a time of the month where testosterone is low. So males, they just have checkpoints, and sperm is continually being produced day after day after day after day. So let's look at the process of spermatogenesis. Now, first, there is a cell called a spermatogonium. Now, this spermatogonium cell is an important cell because this cell needs to replenish itself before it could undergo spermatogenesis. It's important that this spermatogonium divides and replenishes itself, because if this didn't happen, every single one of these spermatogonium, when they divide, if they became sperm cells, these would be depleted. So they need to replenish themselves first, and then they can divide. So there's a backup copy for the next round of spermatogenesis. So spermatogonium divide and replenish themselves. Notice, if these spermatogonium have to replenish themselves before they divide to create sperm, that means as males get older, the spermatogonium have gone through a whole number of cell divisions. And that is one of the major factors why sperm quality declines in older men. Those spermatogonium are copies of copies of copies of copies of copies of copies of copies that are now beginning to divide to create sperm. So after this spermatogonium divides, replenishes itself, it's then going to differentiate into a primary spermatocyte. Now with this primary spermatocyte, this cell is going to be destined to divide into a sperm cell. But remember, this process, spermatogenesis, is a form of meiosis. So this is going to involve 2 rounds of cell division. This primary spermatocyte undergoes meiosis 1. and that's going to result in secondary spermatocytes. Now, with these secondary spermatocytes, they've undergone their first round of cell division, meiosis 1, and now these secondary spermatocytes undergo meiosis 2. When that occurs, they're going to produce spermatotids. Now, notice these spermatids, these look like just regular cells. It's important to note that these cells have a genetic shuffling that has occurred, and these cells, they also have half of the chromosome number. This has 23 chromosomes, this has 23 chromosomes, this has 23 chromosomes, and this has 23 chromosomes. However, these don't yet look like sperm cells. It's because these need to develop further. There is a process where some genes need to be expressed, and the genes that need to be expressed are going to convert these spermatids into spermatozoa, or just simply sperm. Now we have these 4 functional sperm cells. Each of them has 23 chromosomes, and one of them can have 22 autosomes in a Y, 22 autosomes in a Y. The other can have 22 autosomes in an X, 22 autosomes in an X. Each of these has 23 chromosomes. However, they could differ on whether or not they have an X chromosome or a Y chromosome. So it is actually the sperm that determines the biological sex of the offspring. Now let's see how hormones play a role in this process. Well, fortunately, all of the hard work that we've gone through to memorize these hormones will come up again right now, because males have the same structures in the brain, and they produce some of the same hormones that females produce during their physiology. So males, like in females, they have a hypothalamus that releases GnRH. When the hypothalamus in males releases GnRH, this is gonadotropin releasing hormone that's released by the hypothalamus, and it targets the pituitary. The pituitary, once targeted by gonadotropin releasing hormone, this is going to be activated to release follicle stimulating hormone and luteinizing hormone. Once the pituitary is activated, it releases FSH and LH. FSH can target the gonads, or the testes. What FSH does is this specifically targets the Sertoli cells of the testes. Remember, those Sertoli cells are housed within the seminiferous tubules. They're a part of the seminiferous tubules. And the reason why FSH is so important is because once the pituitary releases FSH, that hits the testes, specifically the Sertoli cells, activating them to release androgen binding proteins. Androgen binding proteins, when these are released by Sertoli cells, are going to bind nearby testosterone. They're going to bind any available testosterone in the area and concentrate it, because that high concentration of testosterone stimulates spermatogenesis. To regulate this process, Sertoli cells can also produce a hormone called inhibin. And just like it sounds, inhibin, if too much sperm production is going on, Sertoli cells can release inhibin, and this is going to inhibit sperm production. Now, LH, on the other hand, is the luteinizing hormone. This is going to target and activate the interstitial cells. These interstitial cells are important because once they're stimulated by LH, these interstitial cells are going to secrete lots and lots of testosterone. They're going to secrete a little amount of estrogen and a little amount of progesterone. Notice, males produce very high levels of testosterone. Males also produce small amounts of estrogen and small amounts of progesterone. So the idea that testosterone is exclusively a male hormone and estrogen and progesterone are exclusively female hormones is not correct. Females produce testosterone, males produce estrogen and progesterone. They just differ in the amounts of these hormones that they produce, respectively. So here is an overview. We have the hypothalamus releasing GnRH, targeting the pituitary. The pituitary, once this is activated, this is going to release FSH and LH. This would represent a testicle. Seminiferous tubules contain Sertoli cells. They're stimulated by FSH, and then LH targets interstitial cells. Interstitial cells are cells in between the seminiferous tubules. These are going to release testosterone. That affects libido. That stimulates spermatogenesis. That also provides feedback to the pituitary and the hypothalamus. Estrogen is important for bone density in men. Estrogen plays a role in sperm maturation and concentration in the epididymis. This also terminates growth of limb bones. Progesterone. This is an important hormone because this serves as a metabolic precursor for testosterone and estrogen. Here is one more diagram showing another perspective. It's useful to go over these a couple of times and practice them. See them in more than one fashion, so I've included more than one figure. Green means go. It means it's stimulatory, it stimulates. Red means inhibit. So let's go through this. The hypothalamus produces GnRH that targets the pituitary, and now the pituitary releases LH and FSH. FSH targets the Sertoli cells. This could represent a large Sertoli cell. This Sertoli cell, this large Sertoli cell, is influencing sperm production. This right here would represent a seminiferous tubule. So when FSH is released, a Sertoli cell right at the seminiferous tubule in purple would be stimulated by FSH to release androgen-binding proteins. These are binding and concentrating testosterone so that testosterone can be concentrated and ultimately stimulate this process of spermatogenesis. So here we have these spermatocytes undergoing spermatogenesis. These sperm are developing, and now they enter into the lumen, or the center of the seminiferous tubule. Well, where did that testosterone come from? When LH was released by the pituitary, LH targets the interstitial cells to produce testosterone. Remember, these interstitial cells are outside of the seminiferous tubules, but they are still within the testes. This is where the neighboring testosterone comes from. If too much testosterone is produced, that can send a message to the brain saying, slow down on sperm production. If too much sperm are being produced, Sertoli cells can also release inhibin. That can also act as an inhibitor, saying slow down on sperm production. And that is precisely why, if you were to look at these seminiferous tubules, there's about 250 to 500 of these per testis. They're convoluted, they're coiled, they're very small, and they're very long, and they are lined with spermatocytes. Notice all of these little cells in here are spermatocytes. This big straw-like structure is a seminiferous tubule. This, if you were to cut it, look at it from a frontal view, all of these little dots represent just one spot where it was cut, and you see a frontal view. That is how males can produce 100 to 400 million sperm per day. Here we have interstitial cells in between some seminiferous tubules. So sperm are being produced up and down every single one of these seminiferous tubules. And as sperm are being produced through every single one of these seminiferous tubules, eventually they're going to make their way to the epididymis, and then ultimately to the vas deferens so they can be released. This is showing the process that we covered previously, how spermatogonium are replenished, differentiate into spermatocytes. Primary spermatocytes undergo meiosis I. Secondary spermatocytes undergo meiosis II. These are spermatids that differentiate into sperm. When this process of spermatogenesis occurs, this starts with really cells at the outer portion of the seminiferous tubules moving in. Maturation begins from the outside of the seminiferous tubules moving toward the cavity. So now that we've gone through the process of ejaculation, we've talked about orgasm being accompanied by emission and expulsion or ejaculation. We've talked about the content inside semen and why that's important. We've talked about how hormones influence sperm production in males. Now let's take a look at an actual sperm cell. Let's go through a diagram first. There's not much to a sperm cell. They're very small. Really, they're cells with a nucleus and a tail primarily. A sperm cell has a tail or flagella. This is going to whip in order to propel itself through a female reproductive tract in order to find an egg. That tail is important. Mitochondria is what's going to use fructose as an energy source to beat the tail. The centriole. This is important because this helps generate the flagella. That's where the flagella emerges from. The nucleus is important because this is all the genetic information. I mean, that's the entire point of a sperm cell, is to deliver genetic information. So this has 23 chromosomes, 22 of which are autosomes, and one of them is a sex chromosome, either an X or a Y. Next, the acrosome has hydrolytic enzymes. Now, hydrolytic means digestive. So these digestive enzymes are important. They're inside this acrosome because these hydrolytic enzymes are eventually going to degrade the outer membrane of an egg. Here we have an actual sperm cell, nucleus. You have the acrosome, the acrosome. All of these little circles are mitochondria. And then here we have the long flagella. And not a lot to these cells. Pretty much a nucleus with a tail and a couple of other components. So males are constantly producing these cells. And as I mentioned earlier, because these spermatogonium constantly have to replenish themselves before they divide, they're copies of copies of copies of copies of copies. So sperm tends to decline over time. Let's take a look at some examples of abnormal sperm. Here we have one with one head and two tails. You might think, well, this one may get to the egg twice as fast. Well, unfortunately not. Typically, when this occurs, these flagella, they interfere with one another. So this is actually less efficient rather than twice as fast. Here's one with two heads and one tail. You may ask, well, is that where twins come from? No. Unfortunately, neither one of these would be able to effectively fertilize an egg, and it would be unlikely that this even made its way to an egg. Here we have a big head with a little tail. You say, well, is this the origin of individuals with a high IQ? Well, no. This is because that sperm cell, well, the head, had too much cytoplasm. This will lose its fertilizing power. It will not compete with other cells that are capable of getting to the egg first. Last, a little head with a big tail. Although this might look like it could streamline to an egg and get there efficiently, if it did get there, the reason why it has such a small head is because it's missing a nucleus. Even if this did make its way to an egg, it would not effectively fertilize it because it's missing its genetic information. So now that we've talked about male reproductive anatomy and male reproductive physiology, next week in our next lecture we're going to be discussing sexual and reproductive health in both males and females. And now that we've gone through these various sex organs, we're going to see what happens when they're not functioning properly.